Welcome to the Novus Visual Protocol series. In this video, we'll learn how to perform all phases of a Western blot using the most common methods for this assay. Before we can start preparing the blot, we must first prepare our sample lysate. In this example, we'll prepare a protein lysate from cultured cells. Here, we wash the cells twice with ice-cold PBS and add enough lysis buffer to cover the cells. The choice of lysis buffer depends largely upon the localization of your protein of interest. We scrape the cells and transfer the cell solution to a centrifuge tube placed on ice. In order to solubilize membrane-bound proteins, we will require stronger extraction detergents compared to isolating cytoplasmic proteins. In this example, we are using a standard RIPA buffer, which is a common buffer for obtaining maximum protein yield while extracting proteins from all cellular localizations. It is very important to include protease inhibitors in your lysis buffer, which will prevent degradation of your sample. Always use freshly prepared protease inhibitors, keep samples on ice, and work quickly. We lyse the cells by pipetting up and down, followed by incubation on ice for 30 minutes. Then, centrifuge the cells into a pellet. Discard the pellet and collect the supernatant. This is your lysate. Determine the total protein concentration of your sample lysate by testing a small portion of the lysate with a commercially available protein quantitation assay, such as the BCA. This will assist you in loading equal amounts of protein into your gel. Western blots are traditionally performed under reduced and denatured conditions. These conditions will allow proteins to be separated by their molecular weight rather than their native conformational shape or charge. To reduce and denature samples, dilute each in a loading buffer, such as the traditional Lamley buffer. This buffer contains beta-mercaptoethanol, or DTT, to reduce disulfide ridges between cysteines, SDS to assist denaturing and provide a net negative charge to the proteins, glycerol to allow the samples to sink into each well, bromophenol blue to visualize the lysate, and an ionic buffer. Vortex each sample and incubate at 95 degrees Celsius for 5 minutes to completely denature the proteins. You are now ready to load your samples into an SDS page gel. For this next step, we will separate the individual proteins in our sample lysate based upon their molecular weight using a positive electrode to attract the negatively charged proteins. To do this, we load our previously prepared protein samples into a commercially available polyacrylamide gel. Gels are available in fixed percentages or gradients of acrylamide. The higher the acrylamide percentage, the smaller the pore size of the gel matrix. Therefore, higher percentage gels are better for low-weight proteins, low percentage gels are better for large proteins, and gradient gels can be useful for proteins of all sizes due to their varying range and pore size. Prepare your gel by inserting it into the electrophoresis apparatus and filling with running buffer that is appropriate for your gel chemistry. Rinse the wells of the gel with running buffer and add buffer to the chambers. Load your samples into the wells. If you are unsure of the amount of protein to load, 10 to 30 micrograms of total protein is a suggested starting point, as well as titering the amount of sample loaded. You will also need to reserve at least one well for a pre-stained molecular weight ladder. The latter will allow you to monitor protein separation during electrophoresis and subsequently verify protein weight in your sample during later analysis. Close the electrophoresis unit and connect it to a power supply. Most units typically run 45 to 60 minutes at 200 volts or until the loading buffer reaches the bottom of the gel. During this time, the negatively charged proteins in each sample will migrate towards the positively charged electrode, making their way through the polyacrylamide gel matrix. In this next step, we will transfer our separated proteins out of the gel and into a solid membrane or blot. This is based upon the same principle as the previous step, in which an electric field is applied to move the negatively charged proteins towards a positive electrode. Transfer can occur under wet or semi-dry conditions. Here we will demonstrate the traditional wet transfer method. Start by removing the gel from its cassette, cutting off the top portion containing the wells. Notch the top left corner to indicate gel orientation. Float the gel in transfer buffer while preparing the transfer sandwich. To make the transfer sandwich, you will need a cassette, sponges, filter paper, the gel, and your choice of either PVDF or nitrocellulose membrane. PVDF must first be activated by soaking the membrane in methanol for 2 minutes, but other than this, the choice of nitrocellulose or PVDF membranes is a personal preference. Notch the top left corner to indicate blot orientation, and incubate membranes in transfer buffer for 10 minutes. Create a stack by placing the following components from the black negative cathode to the red positive anode. Sponge, filter paper, gel, membrane. Be careful not to touch the gel or membrane with your bare hands and use clean tweezers or spatulas instead. Touching the membrane during any phase can contaminate the blot and lead to an excessive background signal. Filter paper and sponge. Use a clean roller with each layer to gently roll out any bubbles that may be present, since bubbles will inhibit efficient protein transfer. Lock the cassette and place it in the transfer apparatus containing cold transfer buffer, ensuring that the cassette is properly positioned from negative to positive. In order to prevent heat buildup, it is beneficial to transfer with a cold pack in the apparatus or in the cold room with a spinner bar placed in the bottom of the chamber. Close the chamber and connect to a power supply. Perform the transfer according to the manufacturer's instructions, which is normally 100 volts for 30 to 120 minutes. After electrotransfer of our proteins to a membrane, we will now block the blot, apply a primary antibody specific for our protein of interest, and then a secondary antibody which will recognize the primary antibody. Start by removing the membrane from the cassette and rinsing 3 times in water. As an optional step, we can verify the proteins were transferred successfully by staining the membrane with Ponceau red. Incubate the membrane in Ponceau for 5 minutes and wash with water until the bands are clear. After verification, the blot can then be destained by continuing to wash with water or TBS tween until the dye is completely removed. We need to block all areas of the blot which do not already contain protein. This will prevent nonspecific binding of the antibodies and reduce overall background signal. Common blocking buffers include 5% non-fat dry milk, or BSA, in a TBS tween solution. However, do not use milk when probing with phosphospecific antibodies, as it can cause high background from its endogenous phosphoprotein, casein. Incubate the membrane with blocking solution for 1 hour at room temperature under slight agitation. Decant the blocking solution and wash with TBS tween for 5 minutes. We are now ready to add our antibody. Dilute the primary antibody in a blocking buffer at the concentration recommended on the datasheet, and incubate overnight at 4 degrees Celsius with gentle shaking. A recommended optional step is to also use a positive loading control antibody, which allows the user to verify equal amounts of total protein were loaded into each well and aids in troubleshooting by removing any uncertainties with the Western blot procedure. The next day, decant off the primary antibody and wash the membrane with large volumes of TBS tween and vigorous agitation 5 times for 5 minutes each. These stringent washes are extremely important for reducing non-specific background signals. After washing, dilute the secondary antibody in blocking solution and incubate the membrane for 1 hour at room temperature at the concentration recommended on the datasheet. In our example, the secondary is also conjugated to HRP for later detection. Decant secondary and wash the membrane with large volumes of TBS tween and vigorous agitation 5 times for 5 minutes each. You are now ready for the detection phase. In this final phase, we will demonstrate signal development using the most common, most sensitive, and most inexpensive detection method, the electrochemiluminescent, or ECL, reaction. This method utilizes the HRP enzyme, which was conjugated to the secondary to catalyze the ECL reaction and produce light. The light is then gathered onto X-ray film and developed, or digitized with the aid of a specialized camera sensitive enough for this application. We start by mixing equal parts ECL reagents in a 1-to-1 ratio according to the manufacturer's instructions. We will incubate the membrane for 3 to 5 minutes without agitation. After incubation, decant ECL mixture and use a laboratory wipe to wick off excess solution from the corner of the membrane. Place the membrane in a clear plastic wrap, such as a sheet protector, to prevent drying. Avoid letting the membrane completely dry out. We can now use a roller to push out any bubbles or excess solution. Immediately develop the membrane. Both film and camera systems allow you to adjust the exposure time in order to ensure a picture-perfect Western blot. Relative band densities can now be quantified with commercially available software. Proper molecular weight can also be verified by comparing band sizes to the molecular weight ladder.